pleasure of deciding who to invite for our plenary sessions with my committee and the officers. And I have the pleasure of introducing our next speaker for this afternoon, who is Dr. Jessica Turner, who is, in fact, full professor in psychology and neuroscience at Georgia State University. And congratulations to Jessica, because this has been a very recent advancement. Jessica is a cognitive neuroscientist who collaborates with clinical psychologists and psychiatrists in the study of mental health and function. She received her PhD in psychology from the University of California, Irvine, and did a 2-year postdoctoral position in a non-human primates lab at Rutgers University in New York before moving into clinical neuroimaging. Schizophrenia is her primary research target, having worked with the multi-site collaborative functional biomedical imaging research network at the University of California, Irvine, and at the MIND Research Network in Albuquerque for the better part of a decade. She is currently funded in multivariate imaging genetics analysis applied to schizophrenia, bipolar disorder, and other clinical populations. And students in her lab work on the relationship between resting state measures and schizophrenia, the genetics of brain structure across disorders, and other neuroimaging measures applied to a variety of clinical questions. As I said, she was just promoted full professor at Georgia State University this week. And here she leads the cognitive and affective neuroscience concentration within the psychology PhD program and has a strong commitment to undergraduate and graduate research experience in neuroimaging. And for this, I'm pleased to say that she recently received the Outstanding Faculty Achievement Award for research productivity and graduate student training in research. She's involved in data sharing and reanalysis efforts in the neuroimaging research community with a focus on the semantics of dataset description. As many of you involved in neuroimaging, like me, know, Jessica is the co-chair of the Enigma Schizophrenia Working Group, and she has been in this role since the beginning of the Enigma initiative. And she will be presenting today on its methods and findings. And she will also be around after the talk and tomorrow for any of you who would like to discuss with Jessica the Enigma Consortia initiative and the possibility of getting involved. So I'm now going to leave Jessica to tell you more about this and her work. Jessica.
Well, thank you so much for inviting me to be here today. Those of you who may have been at one of the sessions on Thursday afternoon heard Dr. Nida Jahanshad present a little bit about Enigma as well. So some of these slides may be familiar, but hopefully not too many of them. Enigma stands for Enhancing Neuroimaging Genetics Through Meta-Analysis. Its head, if you will, the instigator behind all of this, is Paul Thompson, currently at USC. The map here shows the current contributors across all of the various Enigma subprojects, showing really the breadth of the contributions around the world. It's obviously not just about schizophrenia, not by any stretch of the imagination, and it wasn't even originally about mental disorders at all. But the innovation, if you will, of the Enigma approach was to say, rather than send me your data and I'll do my own analysis and tell you what happens, the idea was you keep your own data, do the same analysis everyone else is doing, and share the results, so that there's no question of confidentiality or data transfer questions and things like that, insofar as we can stay with a meta-analytic approach. And anyone can participate regardless of how large your sample size is. In imaging genetics, of course, we've realized that we need lots and lots and unbelievable numbers of subjects. And so every study, almost by definition, is underpowered. But if we can each run the analysis and then combine the analysis to see where the consensus effect is, then we can actually get somewhere. And that was really the idea that drove the development of Enigma. So the initial genetics was the first push, looking just at hippocampal volume, intracranial volume. That paper came out in 2012, followed up by a second paper in 2015, looking at all the subcortical volumes with obviously an increased sample size, which led to, again, additional papers on top of that. And the third approach within the genome-wide association studies parts of Enigma is really looking at cortical thickness and area, and as Nada pointed out on Thursday, that is actually available in bioRxiv if you want to go see the results, and I believe it's under review at a journal. I don't remember which one. But this is just an example, of course, of the Hibber et al. 2016 paper on hippocampal and intracranial volume, and the Adams et al. Paper with, hippocampus and intracranial volume for both Hibber and Adams, with 33,000 subjects, which is what it takes to find genetic effects on brain structure. Now, the groups that were participating in these various Enigma studies were not just healthy controls, right? There wasn't just samples of healthy controls. Paul said, send everybody, we don't care. And as a result of some of the initial analyses, there were quite a few samples with schizophrenia imaging genetics in them, and quite a few with bipolar. And so those researchers said, well, why don't we work together and form our own group to try to look at questions that may be specific to our specific disorders? With the result that Enigma now has over 30, I think, different groups that are focused on specific clinical questions. So in the upper left corner here we go. We have the schizophrenia and bipolar, which were sort of the first two to organize back in 2011 or so, 2012, with major depression. And they're working together to try to look specifically now at cross-disorder relationships. There's a relatives group that spun off of schizophrenia and bipolar disorder. Relatively new groups in the last few years is just early onset psychosis, schizotypy, and clinical high risk. Schizotypy will have a presentation later this evening on some of their initial findings. Autism and attention deficit disorder have grouped together, as well as OCD. Suicidal thoughts and behaviors is not really a clinical diagnosis, but they were funded to look across all of the possible groups to anybody who had data about suicidal thoughts and behaviors, regardless of what the comorbidity might be, to see if there's a common effect. And we have the rare genetic disorders and anxiety disorders, and more recently some of the neurological disorders as well, some of which, again, you can see are working together, and then others are sort of on their own. PTSD was just funded, I think, within a few years to work on this. Using, again, a combination of approaches. Most of the groups are using a meta-analytic approach. Some of the groups have decided they really are willing to do a mega-analysis approach, in which case they actually centralize their data and do an analysis that way. But I think most of the groups are still working from a meta-analytic framework. There are also non-clinical groups looking at methodology, because of course, even though we all have a sense of how you do a hippocampal subfield kind of analysis, doing it in a way that is implemented the same way across multiple sites with multiple personnel, and so that it can be explained the same way, so the data curation is done the same way. All of these require a great deal of discussion and testing to make sure that we're actually seeing comparable results across different sites. We also have, of course, beyond just the genome-wide association, copy number variants, epigenetics, and a group looking specifically at the evolutionary marks in the genetics, as well as sex and gender and the lifespan. So there's really quite a lot going on, because Enigma itself is really a generic framework for collaboration, and is not limited to any one particular interest. Usually when a working group gets started, the first thing they do is a subcortical volume analysis and meta-analysis, because that's the easiest one to do. It's the most straightforward one to do. Everybody's done it. We've got some very well-developed scripts and for that. And then from there, they will do a cortical surface analysis, looking at regions, either area, thickness, volume. And by that point, the group's been working together for a while, and they start spinning off secondary projects or questions that may come up that are specific to their particular group or disorder. It's been a very successful approach. As Paul points out, we have 9 of the largest clinical neuroimaging studies, with major depression topping the list with over 10,000 subjects as part of their meta-analysis, with schizophrenia right behind, almost 10,000 subjects, and even with a 22q deletion syndrome study of almost 800 subjects, which is, those of you who work in that field will know, is not the easiest thing to pull together. But when enough people are willing to work together in this research community, we can actually identify these kinds of samples and look at some interesting questions. So I co-chair the Schizophrenia Working Group with my co-chair Theo van Erp, whose picture is up there. We have completed our subcortical analysis and our cortical analysis. We have not gotten anywhere with the genetics. We've sort of been fighting shy of the genetics, but we have been starting in on cross-disorder comparisons with major depression and bipolar disorder. The DTI group, shape group, and laterality approaches have all interacted with the schizophrenia researchers, and we have a number of sub-projects. I'll be telling you about the results of some of them, looking at questions that we're interested in, positive and negative symptoms, the relationships with siblings and first-degree relatives, other characteristics like aggression or the deficit syndrome or cannabis use, etc. I won't tell you about all of them, unfortunately. So our first paper came out in 2015, where we were looking at subcortical brain volumes, and at that point we had 15 sites participating and had almost 5,000 subjects. As we can see with the results here, it's not exactly, earth-shaking stuff, right? We all know going into this that the hippocampus is smallest and the lateral ventricles are increased and everything else is sort of in between. What makes this important is really nailing down these relative effect size. This is what you can expect to see in an international sample of individuals with schizophrenia. And if in reading the literature you have come up with this idea that the caudate is problematic, some studies find it, some studies don't. Some papers show it, some papers don't. There's a reason for that. It basically, even with almost 5,000 subjects, there's no significant effect one way or the other across everybody in the caudate. Those certain subgroups will actually show it. The nice thing about this approach is then in Japan, Dr. Hashimoto and the Kokoro group took the same approaches that we had been using, and in a sample of about 2,000 subjects, used the Enigma approach and managed to replicate the same ordering of effects in their sample, doing a direct comparison, and also they extended it to look at asymmetries left and right. We also, in 2018, just published our cortical paper. It took a few years. The subcortex, everybody could sort of agree on. We had a number of years of arguing over whether or not our free surfer cortical segmentations were really robust enough and reliable enough to do a meta-analysis this way, and eventually we decided that they were, as free surfer cortical segmentation matured and stabilized. And what we see on the left is obviously the differences between cases and controls, taking into account age and gender. Again, this is a meta-analysis across, I think at this point, 30 sites or more. And generally, once you're up to 10,000 subjects, the entire brain is affected. It's just a question of exactly where the largest effects are, which in our case were in the inferior temporal regions, with the rest of the temporal lobe following along behind it, and of course the frontal lobe. Again, as you would expect. The nice thing about where we are now is we've learned to run all of our analyses the first time through, and instead of spending a month arguing about should we include this as a covariate, should we include that as a covariate, just throw it all in there and do it all. So on the right you see the results of doing the analysis, taking into account mean thickness of the cortex, because that's always the question when you're looking at thickness. Should you account for the global mean or not? And the answer is would do it both ways. And in our case, what we're seeing is that large portions of the brain are affected merely in keeping with the mean, but there are regions like the temporal lobe which are more affected than you would expect. And there are regions like the parietal cortex and the motor cortex which are relatively spared. And that was an important thing for us to try to come to grips with. When we look at surface area, on the other hand, we again see the whole of the surface area. Every region has a smaller surface area in cases than in controls. But once you account for the average surface area, pretty much all of it is wiped out with the exception of the precuneus and the posterior cingulate areas. So there's less regional specificity in the surface area effects than what we see in the cortical effects. Now, we can also compare across the different clinical working groups that have done their analyses to date. I think most of these have been published at this point. Because everybody's doing their analyses the same way, we can look at these meta-analyses from disorder to disorder. So in the lower left here, we have the schizophrenia brain, affected pretty much all over. Up here on the right, we have the bipolar disorder, which again, with 10,000 subjects or however many subjects they had, it's the whole brain, but in less of an extent than in schizophrenia. Whereas you can see in the OCD, where again there may be a power issue, relatively speaking, but they're finding a very different pattern of effects. Major depression, not as affected. Alcohol use, very different pattern. And the 22q actually shows a very similar effect in the superior temporal gyrus, but a very different pattern in the rest of the brain. Autism, again, this temporal lobe is affected, but the rest of the brain shows a very different pattern. We have not yet gotten to the point of actually digging into these differences. This is merely for initial comparisons only. We can also look at diffusion tensor imaging. Once we got the structural measurements underway, there were also, within the Enigma methods, a DTI group making sure that track-based spatial statistics could be scripted up and run similarly across multiple sites. And as a result, we had a DTI group within schizophrenia led by Gary Donahoe, whose picture is up there. He's now accompanied by Lorena Holloran working on it. Sinead Kelly, who's one of the travel awardees this year, actually led the paper on the initial DTI working group with about 4,000 subjects involved, which is again one of the largest, or the largest DTI study in schizophrenia, and it was just published last year. Again, with so many subjects, basically the entire, every known tract is affected with reduced fractional anisotropy in schizophrenia, but the important thing is to be able to actually get an estimate of highly reliable effect sizes and the ordering in which these different tracts are actually affected. And in their case, they could also look at mean and radial diffusivity as well as a fractional anisotropy and show that these reductions in fractional anisotropy were associated fairly consistently with increases in mean and radial diffusivity across the major tracts. Besides gray matter and white matter, we can also look at the shape of the volumes of the subcortical regions, because again, everybody was using FreeSurfer. FreeSurfer outputs a fairly standard interface. We could then ask, we know that these volumes are all reduced, generally speaking, except for the ones that are increased, but is the volume equally affected at all points, or are there areas of compression and expansion? And Lei Wang, who was one of the drivers of this project, had a history of looking at subcortical shape going back 10 years or more, looking at effects on the thalamus, but in smaller samples. So now, between Lei Wang and Boris Gutmann, who are shown up here, we have an Enigma shape meta-analysis in schizophrenia. Again, now they have almost 6,000 subjects from different sites around the world, 21 different sites. This required a certain amount of work. to extract the surfaces of the subcortical regions from the free surfer parcellation and co-register it to a shape atlas, and then do all sorts of very interesting computations on it to identify the effects of diagnosis in the presence of age and gender and intracranial volume. And this manuscript is under review at the moment. It's an interesting set of effects because, of course, it isn't a nice little scalar that says bigger or smaller, right? We now have a three-dimensional representation of where these volumes are expanded, shown in red, or contracted, shown in blue-green. And we can see, for example, here in the hippocampus, there are regions that are relatively unaffected and then regions that show strong compressions, or in the thalamus, expansions and compressions sort of combined with each other. I think every region showed reductions, and then some showed expansions as well. Now, besides just the overall case control differences, we are very interested in how symptoms are affected in schizophrenia. What is it that seems to be related to the person's measures of symptom severity or their clinical presentation? The challenge is, of course, in these massive meta-analyses is that everybody used something different, right? These are all legacy data, and individuals collected whatever they thought would be necessary for their particular study. So by far and away, the most common was the PANSS. There was a second cluster with the SANS and the SAPS, and then a third cluster with the BPRS, and then after that, there's just not large groups with very specific kinds of measures. They tend to use very generic symptom measures. Luckily, within the FBURN study that I had been a part of, we had spent some time actually collecting both the PANSS and the SANS and the SAPS on the same individuals with the same raters. And from that, Theo actually published on a conversion between the PANSS and the SANS and the PANSS and the SAPS, so that you could try to predict one from the other. So what Esther Waltons did, shown here, was lead a group proposing again to look at negative symptoms, converting everybody to the SANS, and then also the positive symptoms, converting everybody to the SAPS. There was a lot of interest in this, but at the same time, at the time that she was starting, everyone said, well, we're still not published our cortical paper yet, let's focus on a region of interest. So a lot of the early discussions in these groups was, what region of interest should we look at specifically? And in the negative symptoms, they landed on the medial orbital frontal cortex based on the literature. And in the positive symptoms, they landed on the superior temporal gyrus as sort of the region of interest specifically to look at. And the medial orbital frontal cortex did actually show a strong effect for, well, a moderate effect, not a strong effect, with reductions with increasing negative symptoms severity. Now, the nice thing about the Enigma approach, the thing that I'm most a fan of, is these actual forest plots here, where you can see for every site what the effect size was and the confidence intervals around it. And as you can see, there is a consensus effect that as this region gets thinner, the negative symptoms are increasing, but this is not something that any individual site would probably have published on their own, right? So we're getting at, in a sense, the file drawer problem here, because you would not necessarily see this in your sample of 100 or 200, but when you do 15 of them, suddenly you can get a consensus effect. The superior temporal gyrus also showed an effect, not as strong in the left as on the right, whereas that superior temporal gyrus is thinner, the positive symptoms are more severe. And again, this is not something that any individual site would have necessarily claimed to be significant on their own, but in the consensus, we can see the effect. When we did the overall subcortical and cortical symptoms, where we could look at the whole brain together, subcortically, we did not really see a whole lot with the PANSS. We just saw as the negative symptoms got stronger, the lateral ventricles got bigger. nothing we can do with that. By the time we came up with the cortical analysis, we had learned a bit more about how to do these analyses, and we included those regressions in the original analysis, so we could, instead of looking at a moderator effect, we could look at the individual analyses. And here we see actually that Esther's group was spot on. The strongest effect with PANSS negative is actually in the medial orbital frontal cortex, though we do see when we have the larger sample and we can look at the whole brain that actually much of the frontal cortex is related to negative symptoms, as is the temporal lobe. And when we look at positive, again, the superior temporal gyrus is the top finding, but the rest of the temporal lobe, or other parts of the temporal lobe and some parts of the frontal cortex are related to it as well. We just needed the full sample to really see that. So again, we are in many ways addressing what we might see as arguments in the literature, where some papers find it and some papers don't, some groups find it and some groups don't. The consensus effect is that there does seem to be a relationship in these very specific regions. Subcortically, Boris and Lay also looked at shape severity and found a itty-bitty little spot in the amygdala and in the caudate that seemed to be related to SAPS total. That opens up all sorts of questions about how these relationships relate to the cortical relationships and to other connectivity kinds of measures, but of course, those are questions still to be asked. Within the DTI group, they also looked at symptom severity and they found trends in several of the tracks, but nothing that actually passes correction for multiple testing, even with the 4,000 subjects that they had. So those effects are either not consistent or just very, very mild. However, within the DTI group, they were able to find more than just case control differences. They also went in and looked at IQ measurements across groups. And again, in the literature, it's all over the place in terms of, is there a relationship between white matter and cognitive capabilities or IQ? However, using within the schizophrenia DTI group, using the meta-analytic approaches, again, with a consistent analysis and a consistent way of doing the regressions and the quality assurance, et cetera, they were actually able to find significant effects in patients and controls in both cases separately. Up here we have the healthy controls, again showing sort of the consensus effect, and then the same thing for the patients. And that's a paper, I believe, that is under review at the moment. Of course, we always want to look at duration of illness, and in schizophrenia, that's a very, very tricky question because it is so collinear with age. So across the different Enigma schizophrenia groups, we've had a number of different ways of looking at duration of illness, because we always have to take into account age no matter what. So do we include both age and duration of illness? Do we include duration of illness as a proportion of age? Should we just dump duration of illness and only look at age of onset? What we tend to do is account for age and then throw duration of illness in on top and see what happens. On the right here in the DTI papers, they found no effects of duration of illness on fractional anisotropy after accounting for age. And within our cortical thickness analyses, looking at it region by region, the insula seem to have an increased thickness effect that still existed after accounting for age and gender, but the rest of the brain, basically there was no, once you included age, duration of illness really didn't seem to have much of an effect. However, when we look at each site's mean duration of illness for their patients, and then we look at the case control difference for the overall left hemisphere thickness, we actually see that sites with fairly younger populations, with populations where their duration of illness is on average not that long, show less of an effect than the sites or the studies that were using more chronic patients, even after taking into account age. So there's an interesting cross-study analysis here that we can do, looking at how the sample characteristics relate to the effect size that we're actually seeing. We also, of course, in schizophrenia, you always have to look at medication, right? What are the effects of medication? We know that medication has some effects, but what are they? And again, the question is, how do you measure it? When you're dealing with 50 different studies, some of them only know whether or not their patients are medicated. They're either medicated or unmedicated, that's all you know. Others have every detailed medication that the person was on, but nothing about other medications that they might have been on besides the antipsychotics. Some of them include everything, including chlorpromazine equivalents. So there's all sorts of variability in how you might actually measure medication and try to do an analysis across different studies to see what is the actual effect. The CPZ equivalence is, of course, the first go-to, but there's a lot of arguments about whether or not those are actually accurate and appropriate for the injectables and other kinds of medications. We can also look at the type of medication. Is this person on no medication, a first generation, second generation, or some combination? And then we can also look within a sample or across samples, if this sample is mostly atypicals and this sample is mostly typical antipsychotics, what does that actually do to the effect size as a moderator? So looking, actually, let's start up here, because this was sort of in our initial subcortical paper, where we had no information about the actual levels of medication, but we could look at, for each sample, the proportion of patients who were not medicated at all, because we did have some variability there. Most of the samples, almost everybody was medicated, but there were a few samples where we had, 10% or more were not medicated, and they actually were the ones that showed the largest hippocampal effect size in cases and controls. This was not true across the rest of the subcortex. In our cortical analyses, we actually, again, learned a little bit more about how to do some of these analyses. And so within each site, we usually had a range of subjects who were either who were unmedicated, as well as on typicals and atypicals, or a combination. And so what we did here was actually the meta-analysis of the effect sizes for the subjects who were on second generation or atypical antipsychotics compared to the unmedicated subjects. And they see a reduction in the superior temporal gyrus, the inferior frontal regions, and the superior medial prefrontal regions. In the individuals who are on the first generation or typical antipsychotics relative to the unmedicated, they have a much more severe reduction in this medial prefrontal cortex. So generally speaking, across our, in fact, if you look generally across the brain, the individuals who are unmedicated had the least thinning, if you will, of the cortex, followed by the individuals on second generation or atypical antipsychotics, followed by first and the folks who are on combined, who tended to be have the largest effect sizes in cortical thinning. In the subcortical regions, we're finding certain spots, actually, interestingly, in the amygdala and in the hippocampus for compressions that seem to be relatively, and the accumbens, that seem to be related to CPZ equivalents. So in this case, that's how they actually looked at it across those patients that had that information. So I want to sort of stop there for a moment and talk about what we've really learned from the Enigma so far. Right, from the Enigma Schizophrenia group. One thing that really hit home is, one study is never enough. One paper is never enough. When you look at these forest plots, what we've got here is the hippocampus on the left, and I think the strongest cortical effect on the right, from the Enigma Schizophrenia papers. And those are the findings that are fairly consistent. Almost every site finds a smaller hippocampus in their individuals with schizophrenia. Not everybody finds it to be significant, but everybody finds it in that direction. And the same for the inferior temporal regions. They find a reduction in the schizophrenia patients. However, when you're starting to look at more nuanced effects, such as the relationship between brain structure and negative symptoms, then you really need 15 sites to get a sense of what's going on. You can't just look at 1 or 2 or even 3 sites to say, well, it's significant, therefore we're good, or it's significant, we've managed to replicate it. No, we need to actually get a sense of what the consensus is and what the power is to really see it. And of course, with multiple studies, you can now start to do the moderator analyses, as in the hippocampal studies, to say, what is it that's really driving the differences across the different studies, rather than speculating when you look across papers and saying, well, this group was younger and this group was slightly more male. Does it really actually make a difference? When we actually start pulling every sample together, we can start to see whether these things make a difference. So there are pros and cons to the Enigma approach. Some of the advantages, of course, as I think of it, is it's sort of an instant replication, right? Rather than doing one study and then publishing it and waiting for somebody else to collect the data and do a similar analysis and then try to get a replication of studies published somewhere, which journals don't always look favorably on. It's an instant replication where you can look across multiple studies and see whether or not you're seeing the same effects. I put instant in quotes. Is nowhere near instant. It takes months to get everybody, if not years, to get everybody to do the analyses. Remember, we're doing this largely on a volunteer basis. It's as people have the time and the ability to run these analyses and to send back the results and do the meta-analysis. So it's instant in the sense that we all get it done eventually. This replication and this kind of approach of everyone agreeing to try the same things on their data can be a safeguard against false positives in the literature, right? It can also be a safeguard against false negatives, as we've seen. An additional advantage for those of us who are very interested in the replication and reproducibility crisis is that the analyses tend to be recorded very carefully. We have to be able to explain to every new postdoc who shows up or every new graduate student how it's done so that each site is comparable from one to the other. And this leads to levels of detail you would not necessarily see in either a presentation or in a publication that make it really quite reproducible from time to time and from replication to replication. We also, through this method of combining analyses, can lead to very large-scale samples that are necessary for the small effect sizes. The genetics is, of course, the sine qua non of small effect sizes, but in the cases where we're looking at the effects of cannabis or aggression or something else, then of course we're also looking at small effect sizes. And a very promising result from these Enigma approaches is that the multi-site heterogeneity was not a showstopper. Right? There was a lot of argument that if you're trying to look at effects from a 1.5T and effects from a 3T, never mind the differences between the different ways of collecting the data, that the heterogeneity would swamp the effects. And the fact of the matter is, when we're doing a meta-analysis, it doesn't seem to swamp the effects. We can see consistent effects from site to site, scanner to scanner, protocol to protocol. So the multi-site heterogeneity is particularly amenable to the multi-site meta-analytic kind of approach. Now, I wouldn't want to necessarily combine patients from one scanner and controls from another. That's out of the question. But when you're trying to do a meta-analysis of the effect size within a scanner, then that is certainly appropriate. And through explicitly doing cross-study comparisons through modulation, we can look at how the different study samples and different study characteristics can affect the analyses. So in some of our first papers, we explicitly looked at, do we see differences in effect size between 1.5T and 3T? Do we see differences in effect size by age, by mean age of the sample and things like that, which can be very informative, very reassuring, or open up all sorts of cans of worms, depending on what you actually identify. It's a very collaborative approach. So we're getting investigators from around the world talking about how they do things, what seems to work, what covariates they think are really critical for including in any particular analysis. Across the different working groups. The working groups that have started up more recently have certainly learned from our mistakes over the past 5 or 6 years so that every analysis gets slightly better, slightly more complete. These standardized approaches being used across the different working groups can make the comparisons much more meaningful in a way that's incredibly valuable to the research community. And there's obviously a growing enthusiasm for this approach that is reflected in the different disorders and methodology groups that are just burgeoning and showing up faster than we can keep our website updated to keep the list up of what's actually happening. It's something that seems to tap into how we as a neuroimaging or a clinical neuroimaging community can work. So that's all the advantages. Of course, some of the disadvantages are we have large-scale samples as long as everybody has the same measurements, right? So we're a little bit limited in what we can actually ask for. You notice we go for the PANS, we go for the SANS. Nobody has 2,000 samples with the SIRATs, right? Which is, hallucinations are what I'm really interested in. But we have very small samples for those more specific kinds of questions. With 10,000 subjects, that you can sometimes find 200 or 300 that might have your particular questionnaire, and that can be very useful. Or there's been a lot of work done in trying to harmonize across different cognitive measures or different cognitive batteries or different symptom scales, and that can be worth the effort. But we don't have a lot of interventions. We don't have a lot of what in more rigorous communities we might call confirmatory analyses, right? We're looking at exploratory analyses. Even if we have a hypothesis, which we often do, we often say, here's explicitly what we're looking for and why we're looking for it and why we think it's there, we're working with data sets that were already collected. We're not doing the extremely rigorous approach of starting with a hypothesis, designing our experiment, collecting the data for that experiment, and then publishing on it. So these are legacy data. You have to analyze what's already there and make the best of it. But at the same time, we can do quite a bit of that. And there's a lot of questions that are certainly worth asking from what we already know about these disorders and the kinds of measures that have been collected. So there are ongoing projects within Enigma Schizophrenia that I haven't been able to say anything about. They're still in the works, and they're looking for groups to join in. So within the schizophrenia DTI group, they're moving into a different analysis of symptoms and symptom severity. The laterality analyses are still ongoing, trying to look at asymmetries across cases and controls. The aggression subgroup is trying to look for measures of hostility and aggression and how that relates. There's a deficit syndrome subtype analyses, which I think is actually in the manuscript in preparation. They may not still be collecting data. There's analyses looking at sulcal shape and depth rather than volumes and thickness. There are groups looking at childhood trauma for the subsets of the larger schizophrenia groups that have childhood trauma questionnaires. They're trying to look at how that is related to structural measurements. Brain age is all the rage these days, and there's certainly a brain age analysis within schizophrenia to see whether or not predictors of brain age are affected by the disorder. Hippocampal subfields is an analysis. There is actually a group looking at algorithmic approaches to try to reduce these site differences using the Combat software to see if that makes a difference. And there's, of course, explicitly cross-disorder work going on with bipolar disorder and major depression disorder. Though, of course, we have a lot of work to do looking at how do you account for medication differences when every group is on a different medication? How do you account for duration of illness when bipolar disorder has a particularly fluctuating pattern as compared to schizophrenia? And questions like that. We're still discussing how to do all those analyses. There are related groups that interact with the Enigma Schizophrenia very closely. The early onset psychosis is looking for studies where the individuals were particularly young when the psychosis started, and that the scans were done at a fairly young age, I think in adolescence. Enigma Schizotypy has its initial results out, but is always looking for more individuals to participate. And the Enigma Clinical High Risk, I believe, is also looking for more groups to join in. And the Enigma relatives, I wanted to give them a shout out because their paper was just accepted at Biological Psychiatry. Congratulations to them. With 6,000 subjects and 34 different groups participating, looking at familial risk between schizophrenia and bipolar disorder. I won't steal their thunder and tell you what they found. You have to go find the paper. We've got a lot of new directions to go in. Within the DTI particularly, we've been focused on track-based spatial statistics, so sort of a voxel-wise fractional anisotropy measure, and of course everyone wants to do tractography. But as you might know, tractography is very hard to do reliably across different individuals and different groups. And so that is an ongoing methodological question of how do you actually get a tractography pipeline all buttoned up and shared using either Docker or some other kind of form across different groups so that it can run reliably. Also, every analysis that has been done to date is a one-shot regression, right? Stuff you can do in SPSS. How do we actually do the more complex iterative analyses such as machine learning approaches, or of course I'm a fan of independent component analysis? That cannot actually be done the way we're currently doing Enigma. However, we are trying to develop distributed analyses and distributed frameworks where Enigma can be automated. Right? So as soon as you sort of line up and annotate your data and tell us how it's all set up, nobody personally goes in and touches your data, but basically a little bot will go in, analyze the data, send it back, get the feedback, do it again, and whatever it needs to do in order to do these kinds of iterative convergent analyses. There's a lot of promise in that direction. So, and finally, we're not a closed shop. Right? If you think all the papers are out and there's no reason to join in, I want to encourage you to get in touch. Even if you only have 20 individuals with schizophrenia and 20 controls, we're happy to talk to you. You don't have to be a site with hundreds and hundreds of subjects in order to participate. So please get in touch if you want to contribute. I get a lot of people and particularly computer scientists and the computational folks saying, you have a lot of data, can I have the data? And the answer is no, because I don't have the data, right? The whole point of Enigma is that I don't have the data in a centralized repository. The data is with each person. So if you want to do a new analysis with all of these sites, you need to propose the analysis and then work with all of the sites to actually get the analysis done. And we usually strongly recommend that anyone who wants to come in and say, oh, I have this great idea to do an analysis with the Enigma data, to work with some of the other ongoing projects and see how it's actually done and participate before you step up and ask everybody else to do a whole lot of work for your particular analysis. So there's a give and take involved here. We do have a memorandum of understanding. Currently it's about 6 pages long. I say it's about 4 pages of how to play nice, right? How to actually work in this kind of a collaborative environment without scooping each other, without stealing ideas. Just, do unto others as you would have them do with your data. There's a couple of pages of data protection statements for those projects where they actually do try to share data. There has to be some explicit statement that when you receive data from somebody, you're doing one analysis. They're not giving it to you in perpetuity to do any analysis that you want to do on it, and there's a lot of interaction between you and wherever you got the data from. But it's not really too bad. So in conclusion, this is probably not news to anybody who's in this room, but big data techniques have reached clinical neuroimaging. It's not as big as perhaps the geosciences, but from the point of view of those of us who are used to collecting 20 or 30 subjects, 10,000 is big data. These analyses, and this is something I'm very clear with for my graduate students, is these are being done by teams, interdisciplinary teams that require clinical expertise, statistical expertise, imaging expertise, computational expertise, data management expertise. It's definitely going to be a large effort. However, these large-scale collaborations, and I want to highlight, these are volunteer collaborations. Enigma, the central group has gotten some money, but the Enigma Schizophrenia was running long before there was any money involved, and the same for bipolar and others. And we still can answer some of these questions fairly definitively, which means the rest of you don't have to worry about asking about hippocampal size in patients anymore. Examining cross-study differences in samples explicitly, such as a proportion of a sample that is unmedicated or the mean age of a sample, can actually improve the reproducibility, reduce the heterogeneity of results, and improve our understanding. And if you can run analyses on your neuroimaging data and you want to share the results, or you're willing to share the results, then we want to hear from you. So that's the whole thing. There are loads and loads of acknowledgments. This is actually Paul's slide identifying 900 researchers from around the world involved in Enigma at all levels. Funding is too much to speak to because it's basically every group has gotten money from somewhere in order to do some of this. And we have a lot of people who are involved, from undergraduates and graduates on up to senior researchers and other personnel. So with many thanks to everybody who's been involved, and thank you for your attention.
We have time for a few questions. Yes?
Hi, Jess. Great summary of all the great work that you guys have been doing. As a proud member of Enigma Consortium, I actually was surprised that I was wondering if you could address sort of a flip side of one of the pros that you mentioned. So one of the pros that you mentioned is that you have a really big sample size so you can find small effects. The flip side of that is that you can find small effects, but are those small effects meaningful? What does it help us to know that there's a really small relationship between superior temporal gyrus and negative symptoms, and does that have clinical meaningfulness, and what is your thoughts about that?
Yeah, that's always the question, isn't it? If it takes 10,000 subjects to find the effect, do we really care? And I think there's levels of answer to that question, of course. I mean, one is, if somebody walks into your clinic, are you going to scan them and look at their superior temporal gyrus? No, you're not, right? That doesn't play that level of a role. But on the other hand, can it give you a clue as to the mechanism, as to the issues? Does it play a role in the convergent evidence between what is the role of the temporal gyrus and prediction error or whatever model of schizophrenia? Absolutely. So I think it depends on the level of the question as to how important that effect needs to be.
Yes, one question here.
This is sort of a follow-up to the last question, which is actually I was really grateful to see those effect sizes. They told me I didn't need to do structural imaging to find relationships to symptoms, and that this, whatever there is, I think the brain does have something to do with the symptoms of psychosis, but measuring structure probably isn't the go-to for doing that. So that brings to mind other sorts of modalities of imaging, and you didn't say anything about, you spoke about DTI, but of course there are. Could you say more about how Enigma is working with other modalities? We discussed earlier functional modalities, but of course there are others as well.
Yeah, that's an interesting point. I don't know if we have an Enigma PET consortium, and I don't think we have an Enigma ASL consortium, so anybody who wants to start one, it's wide open. We do have people looking at resting state. Peter Kochanoff is leading the resting state group and just published, I think, quite recently, a paper validating a pipeline that is going to start to roll out to various groups. So you'll notice at the moment I had no Enigma schizophrenia resting state, and everybody wants to do it. So as soon as there is a pipeline that everybody sort of believes and agrees on and can implement, then that will be one of the next things to come through. In terms of task-based analyses, of course, everybody has their own cognitive task. So we don't have 10,000 subjects with the NBAC or anything like that, though there could certainly be subsets within Enigma of people saying, well, if I have an auditory oddball and you have an auditory oddball, let's do a meta-analysis and see what's going on. Again, it's completely driven by the research community, so if somebody wants to step up and lead that, that'd be absolutely fine. Is that what you meant? Basically, good.
Any more questions? Okay, well, Jessica's going to be... Oh.
I think I was kind of caught up by the fact that, like you point out that a lot of the finding you can find on the big, large sample level is not really seen in the original small studies. And you also point out that a lot of the data, like acquisition kind of parameters, are not the same. And it kind of reminded me, like when I learned about meta-analysis as a student, like there was this criticism that meta-analysis can be like garbage in, garbage out approach. And I think Enigma definitely have a lot of mechanisms to like really prevent that. But I'm also very interested in do you think Enigma findings should be held up to replication? And if that should happen, how would that be? Like, how can you find another sample size this large?
Exactly. Yeah, it's an interesting question. I have to admit, once I've got 5,000 individuals with schizophrenia, I don't really want to replicate that for these imaging studies. And I think the question, that's one of the reasons I find the forest plots to be so useful, and the measures of heterogeneity across the findings, across the results, is because you can sort of think of those as either replications or failures to replicate and get a sense of what is sort of the average finding if you were going to look at it, which gives you your best predictor of what would happen if you did your own study. I don't know. I tend to think of meta-analyses as something that don't need replication per se. Maybe they need updating in a few years, like as our neuroimaging techniques change or the medication takes a complete change, we might want to do some of these analyses again. But I certainly wouldn't prioritize the replication of these efforts, not like the subcortical analysis, right?
Thank you.
One question.
Hi, that was such an interesting talk. Thank you. Speaking of replication, because you're finding effects of medication versus no medication, and yet you point out that the data on medication is inconsistent, and probably doesn't include things like, is somebody off medication just right now, or were they medication naive? It occurs to me that this data may need replication with medication naive individuals to really be able to figure out what part of this is medication, and then ultimately, if some of it's medication, then what medications are we talking about, at what dose, and for how many years, et cetera.
Absolutely.
So I think your work is still cut out for you, I'm afraid.
Oh yeah, no, yeah, definitely. No, I have to agree with you. Medication is one of those things where we're not doing a clinical trial, right? We're not testing on and off. We're literally looking at what's out there and doing the best we can with the information that we have available. So we have, in a sense, promissory findings in fairly large samples looked at from a number of different ways across different modalities. But certainly we can't tell you about risperidone versus olanzapine. Just can't do it.
Right, and you can't tell us about, never risperidone and never olanzapine versus not at this moment versus this moment.
And we can't, in many cases, one of the reasons I sort of shied away from the chlorpromazine equivalents is because we may know what the doctor told them to take, but we don't know if they're actually compliant. So we're guessing on that as well.
Yeah, thank you.
Hi, Jess. Very nice to see this overview of the studies that I know very well, but still it's good to see them all in one story. Following up on the medication issue, I also think that this medication might reflect a lot of outcome of the patients, because the ones that are not on medication probably doing very well.
Are doing rather well, right.
And we haven't corrected these analyses.
Yeah, we did try to, actually, Theo did quite a bit of work in the cortical analysis looking at CPZ equivalents, correcting for symptom severity. And those are, again, buried in the supplemental material that I don't have at the top of my head right now. But yeah, you do need to tease apart what's the relationship between symptom severity and medication, but also you don't know what they were before they were medicated, which is really the question. Yeah.
Yeah, so for the medication effects on the brain, I think the jury is still out there.
Oh yeah, definitely.
Okay, well, Jessica, thank you very much. On my side, I can say also as a member of the Enigma consortium, one of the last things Jessica said is that it is truly a really nice group of people to work with. It is a very well-behaved group of academics. So if you want to ask more questions to Jessica, she will be around, as I said at the beginning, and join me in thanking her again. Not just for this, but for the Enigma.